The outermost layer is epiblema. Many of the cells of epiblema protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as the radial walls of the endodermal cells have a deposition of water-impermeable waxy material superin in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. As compared to the dicot root, which has few xylem bundles, there are usually more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous roots do not undergo any secondary growth. Dicotyledonous stem. The transverse section of a typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem, covered with a thin layer of cuticle. It may bear trichomes and a few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of collenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of monocotyledonous stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles. It's surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundles. Dorsiventral dicotyledonous leaf. The vertical section of a dorsiventral leaf through the lamina shows 3 main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface adaxial epidermis and the lower surface abaxial epidermis of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called mesophyll. Mesophyll with isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both the surfaces of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermal cells along the veins modify themselves into large, empty, colorless cells. These are called puliform cells. When the puliform cells in the leaves have absorbed water and are turgid, the leaf surface is